Hi, everyone. Welcome to our ICRN Translational Seminar Series. We're delighted that everyone is joining from all parts of the world, I hope. Today we have the great pleasure of having Dr. Gauthier Bouche. I hope I didn't butcher your name. Usually I'm at the receiving end for these kinds of things. Dr. Bouche is joining us today. He is a medical director for the AntiCancer Research Fund. They are doing a number of exciting endeavors, and the topic that we wanted to gain his insights on today was a very interesting one with regards to impact, which is instead of developing new drugs, which can take a long time and be an expensive process, how could one think of ways to repurpose existing drugs, which are already out there for many other diseases and are hopefully quite inexpensive, so accessible to people in regions of the world where cost may be a barrier. So we'd love to hear Dr. Bouche's thoughts today on this. Welcome.
Thank you very much, Mitesh, for the nice introduction. You got my name properly, except that it should be Bouche and not Boucher, but that's all right. Thank you very much for the invitation. Thanks so much for allowing me to present the data we have gathered about the repurposing of drugs for cholangiocarcinoma. I will now share my screen and give my presentation. I hope just a minute. So it will enter full screen. So it is now. So I'm working for the AntiCancer Fund in Brussels, in Belgium. We're a foundation and not-for-profit organization, and we support clinical trials mainly. We are interested in interventions that actually have no commercial prospects, and so because of that, we became quite quickly interested in the repurposing of existing drugs. But this is not the only intervention we're interested in, for your information. So I don't know if everybody's very familiar with drug repurposing, so I thought it would be good to introduce a bit what we're talking about. And so there's a good definition published last year, but the strategy. So it's a strategy for identifying new uses for approved drugs that are outside the scope of their original indication. In their definition, they have also included experimental drugs, but as a non-profit organization interested in intervention with no commercial prospect, we limit the definition to only the existing drugs. So the rationale for drug repurposing is that we all know, and the clinicians attending that webinar are probably aware of that, the list of secondary uses of drugs is quite long. Surprisingly, there's not a lot of quantification of that. And I'm only aware of a couple of examples, which is one in pediatric hemato-oncology, where Julie Blatt of the University of North Carolina has identified 39 drugs that had been repurposed from other indication to pediatric hemato-oncology, which is quite a lot. There's also a very interesting exercise done by von Eppel at the MIT. They looked at 29 drugs approved for the first time by the US FDA in 1998, and they interrogated a drug compendium called Mitromedex for 5 years later, just to see if there were new uses suggested by that drug compendium. And you can see that 143 new uses for those 29 drugs were recorded in that database in 2003. So it shows a bit the extent of and the potential of drug repurposing, though there's still some need for better quantification of that. I think it's even more important for rare diseases and rare cancer, because we know it's very hard to motivate commercials, drug developers to develop new drugs in rare indications. When I introduce our work, I often get rather a critical comment and skeptical comment about the fact that there's not a lot of repurposing successes in oncology. And beside thalidomide, not everybody will remember other examples. So I want to, with that slide, explain that there are multiple examples, and it's important to understand that there is hard repurposing, meaning that you have a non-cancer drug that you will repurpose in a cancer indication, and to know that there is also some sort of soft repurposing. So you have a cancer drug that you want to use in another cancer indication. Actually, I don't like that binary classification. I prefer to see it as a spectrum, because sometimes in soft repurposing as well, the indications are quite different. So here, there are a few examples. BCG moved from prevention of TB to bladder cancer, quite a big step. Then you know the story of thalidomide, all of you, I'm sure about that. The steroids are not usually seen as a successful example of repurposing in oncology, but I think it is. It went from anti-inflammatory and it's extensively used now in hematological malignancies, and in particular in acute lymphoblastic leukemia. Zoledronic acid moved from osteoporosis to the prevention of skeletal events of bone metastasis. Imatinib moved from hematological malignancy, CML, to sarcoma, which is also quite a big step within oncology. And I used the trastuzumab example a bit as a provocative example, just to show that even within oncology, you have some repurposing going on, and it's usually not coined as such. But trastuzumab was clearly developed for breast cancer, HER2 positive breast cancer. And then at some point, we realized that gastric junction cancer, at least a substantial proportion of them were expressing high level of HER2. And so we could reuse that drug, which would have never been developed for that specific indication, to treat those cancer. Now, as a foundation, we believe that it's important to invest our money, time, energy in repurposing for several reasons. And I use the analogy of the toolbox. I think the drugs we have are a great toolbox. So we know we have more than 2,000 drugs ever approved worldwide. And the nice thing about that toolbox is that we get approximately 30 new tools every year. So this is a growing toolbox. The nice thing as well about that toolbox is that some of the tools can do several things. So this is called polypharmacology, which is usually not seen as something very positive by clinicians. But in terms of drug development, I think it is very positive, at least in some instances. So we know that drugs usually have multiple targets, or then they can eat the same targets in different tissues or on different cells. And even for monoclonal antibodies, which we think are very specific, in the end, we realize as research goes and science advances that those monoclonal antibodies can also be reused. For example, rituximab, which targets CD20, has also been shown to target a specific molecule on podocytes on the kidney, and that has been used therapeutically in some kidney diseases. So this illustrates that science will keep on shedding new lights on diseases and drugs, and that we can look in our toolbox and find new ways of tinkering. And so to apply that concept of a toolbox to cholangiocarcinoma, I decided to look for recent papers in cholangiocarcinoma, and almost randomly, I picked this one. So that's a JCI paper from a very good group at the Mayo Clinic. And this was supported by the Cholangiocarcinoma Foundation, by the way. And they showed that for anti-PD-1 to be effective in cholangiocarcinoma, you need to target as well the tumor-associated macrophages and the granulocytic myeloid-derived suppressor cells. To do that, they have used pharmacologically mouse antibody against CSF1R, which will target tumor-associated macrophages. And they have used a small molecule called GW3965 to target as an agonist of liver X receptor, which is the target of the granulocytic myeloid-derived suppressor cells. So as you can see on the right-hand side of my slide, that's a combination that you can see on panel D in turquoise has been quite effective as reducing tumor weight with anti-PD-1, anti-CSF1R, and the small molecule against liver, the agonist of liver X receptor. And it's also very effective to deplete PD-L1 positive tumor-associated macrophages, something you will not be able to achieve with anti-PD-1 alone. At the end of that paper in the discussion, they are talking about the clinical translation of their work, and rightfully, they find that they are CSF1 receptor antagonists that have cleared phase 1 and could be used. Or an alternative would be to use some of the drugs that have been proposed to deplete tumor-associated macrophages, and that has to be done together with a liver X receptor agonists such as RGX-104 that are also clear phase 1. So in my opinion, and I'm sure a lot of you will agree, the clinical translation of that work will not be straightforward. We know that the pharmacological inhibitors they have used in mice will not be possibly used in human, or at least not very soon. And even in the combination they propose in their discussion is a 3-drug combination that combines an anti-PD-1. I think access to anti-PD-1 might be possible and actually quite easy, at least in Western countries. And then this should be combined with 2 experimental products from 2 different companies. And so those of you who have worked in clinical trial know that trying to convince 2 different companies to combine their drugs that are not yet on the market is not an easy goal. So this, to me, will make that translation very complicated. So I think that a combination of 3 approved drugs would make things much easier, but still challenging. I'm not disputing this. But I looked into the toolbox. I didn't use any magic trick. I just looked into the literature and into drug bank, drug databases. So I could find that there are existing CSF1 receptor inhibitors, and those are tyrosine kinase inhibitors that you are very familiar with, such as imatinib and sunitinib. So this was published by a group at Abbott Laboratories because they wanted to repurpose one of their tyrosine kinase inhibitors. But in that same paper, you see that imatinib and sunitinib at decent concentration are very potent CSF1R inhibitors. Against the tumor-associated macrophages, the very interesting review from Caseta also indicates a few drugs that can do that. And one of them is clodronate, which is an approved biphosphonate. And I mentioned this one in particular because this will come back later in my presentation. As a liver X receptor agonist, there's a group at the University of Pittsburgh that has shown 10 years ago that actually lansoprazole, again, at a low concentration, was a liver X receptor agonist. So why am I saying all this, not to suggest that Leuyard and colleagues should have done that, but I think it's very important when you do such very interesting work to think of the clinical translation, and maybe some additional work could have been done with those drugs, for instance, just to assess whether they could confirm the efficacy of that strategy using drugs that would have an easier clinical translation. This leads me to the main topic of my talk, which is what we have found in the literature about cholangiocarcinoma and drug repurposing. So I'll present our methodology and the main results that we have found. This work was done a bit more than a year ago for the Cholangiocarcinoma Foundation. We wanted to do that collaboration, and I have updated it with the help of my colleagues a couple of weeks ago for with the aim of that presentation today. So to explain what we have done, I have to introduce one project we started a long time ago, 7 or 8 years ago, which we have called the Repurposing Drugs in Oncology project, or REDO project. This is a very simple exercise. We basically decided to list all the non-cancer drugs with at least one article reporting anti-cancer activity in PubMed. So any anti-cancer activity in vitro, in vivo, against any type of cancer, just to have a very broad list of drugs. That list is available online on that website. And we started to collect data on those drugs, basic information such as the pharmacological class they belong to, the main indication, whether they are on the list of the World Health Organization for essential medicines, the patent status, the type of publication, and the known molecular targets of those drugs. When we did the updates a few weeks ago, we had at that time 322 drugs on our redo list. So we did basic queries for that on PubMed and on the clinical trial database registries, not only clinicaltrials.gov, but also the one from the World Health Organization that incorporates registries from different countries. We screen all of the abstracts and the clinical trials coming from those queries. They were reviewed by myself or some of my colleagues for their relevance to cholangiocarcinoma. And we collected additional information on those abstracts, in particular the type of evidence that was mentioned in the abstract. Then there was a more subjective exercise about the selection of the most interesting candidates that I will show you. And of course, we didn't stick only to that methodology, because any finding that would come accidentally would of course be integrated, because the goal is to inform future research and not only stick to a very strict methodology. Any previous knowledge by any of the members of our team could also be integrated at a later stage. So this is the flowchart of our queries. In PubMed, we started with the 322 drugs. This brought 556 abstracts for 118 different drugs. But of course, not all of them were relevant. And so after the screening, we ended up with 163 relevant abstracts that were about 58 different drugs. We did the same exercise for the different clinical trials registries. We got only 31 trials, but out of those trials, only 11 were relevant, and they actually were covering the 10 different drugs. So this is another way to recapitulate the results. You see that out of the 58 relevant drugs, there were 18 with some human data. So could be observational studies, could be case reports, could be clinical trials. We had 17 with only in vitro data, which is not very encouraging, because we know in vitro is certainly not enough to move things forward. But we had in total 41 with more than just in vitro data. In terms of trial, as I said before, we had 10 different drugs, and actually one of them was not included in the 58 that we had found on PubMed, meaning that there is a trial in cholangiocarcinoma with that drug, but we could not find in the literature any data supporting their use specifically for cholangiocarcinoma. Doesn't mean that this is not relevant. There could be extrapolation from other cancer types, of course, or unpublished data. This is a list of the 58 drugs. I'm not going to name them one by one because I'll make a lot of mistakes, first of all, but also because I know you will have access to that list later on. So if there's any drug you want to look into, you will be able to look into each of them. So what I will do is I will extract, I will focus on a few of them. But before doing that, I wanted to list as well the trials that we have found. Again, I'm not going to go through each of them, but you've seen I've put some comments on my slide for each of the trial. Just want to highlight the three ones, the three in green. There's one trial with the combination of metformin and chloroquine ongoing and actually completed in the Netherlands. There's no results yet. It was for all tumors with IDH1/2 mutation, and that included cholangiocarcinoma. There's an interesting trial that is coming from our updates since last year, with called Shishin. It's a single-arm trial that is registered quite recently in Taiwan, and they are going to include patients with hepatocellular carcinoma or intrahepatic cholangiocarcinoma. This is based on the preclinical data from the same group. The last one is a trial in China with lefamisole. It's a Chinese randomized trial, which will actually assess the efficacy of lefamisole in addition to enlotinib. Enlotinib is a tyrosine kinase inhibitor approved in China, and it targets VEGF. There's no published data, but in the trial registration, they mention data of their own that is unpublished. So now I'll list a few candidates that we found interesting. I'm not saying that those should immediately move to the clinic, but I think it's important to share the result to inform future research with those drugs. So the first one is clodronate. Clodronate, as I mentioned before, is a biphosphonate. It's off patent, and it's approved for prevention of skeletal events in patients with multiple myeloma and breast cancer. Preclinically, it's broadly used in liposomes to deplete macrophages. It works very well to do this. So it depletes macrophages very effectively in animal models. It would probably be useful to use that liposome formulation in trials, but it's not approved as such. There has been trials done with a liposomal formulation in humans in arthritis and in dogs with soft tissue sarcoma. So it's probably possible to run clinical trials with liposomal formulation, building on the previous experiences from the different academic groups. For your information, there's some patent by Stephen Doe at the Colorado State University. But the main paper supporting the use of clodronate applied to cholangiocarcinoma is coming from Edinburgh by Bolter in the JCI 5 years ago. They've shown that it was able to reduce proliferation and increase apoptosis. And as you can see on the right-hand side, you have a very much lower volume with the lipiclode, so the liposomal formulation. In two intrahepatic cholangiocarcinoma cell lines, but it didn't work in one extrahepatic cell line. They have nice mechanistic working of this, and they show that it's related to Wnt signaling inhibition. A second interesting drug is Ivermectin or Ivermectin. It's off patent. It's an antiemetic. It's approved for river blindness. The problem with that drug from a clinical standpoint, it's usually used short term, like once a year on a regular basis. So we have limited knowledge about the long-term exposure of this, though there is development against malaria ongoing with long duration of treatment. So I guess those trials will be informative for the long-term use of ivermectin that could be also applied to oncology. The supportive evidence, preclinical evidence, is coming from Japan in a PNAS paper where they showed that ivermectin was able to reduce tumor growth in 2 different models. And so you can see on the right side that ivermectin was working alone, but was even more effective in combination with doxorubicin. They suggest that it's working through inhibition of YAP1 signaling. I want to emphasize, because I know there is clinical trial ongoing with the zatinib and other tyrosine kinase inhibitor also targeting YAP1 according to a drug screen done in the literature. A third example is the example of Genitidine, which is of patent, which is also of patent. It's a histamine 2 receptor antagonist. It's approved to prevent and treat gastric ulcer. It has a very good safety profile, and you can go with very, very high dose, as it's approved as well for Zollinger-Ellison syndrome. In a paper from 2 years ago, they have shown those data that you can see on the right side. It was able to reduce proliferation and angiogenesis, was able to reduce tumor growth in one model. They've shown that it was related to a direct antagonism to H2 receptor that are highly expressed in cholangiocarcinoma. So I think that's really interesting. And they could show similar results with another histamine receptor antagonist, but this one targeting histamine 1 receptor. There are interesting results with the angiotensin receptor blockers losartan and telmisartan as well. Those are, again, of patent drugs used for hypertension. Both drugs are usually they are grouped together because they belong to the same pharmacological class, but they are very different in terms of pharmacological properties. And telmisartan has some additional advantage over losartan because it's a PPAR gamma agonist. There is very interesting work being done in Harvard on pancreatic cancer, and I'm sure some of you are familiar with that, with losartan. And as you can see, there were 2 papers quite recently from Japan with in vivo data suggesting that both of the drugs could work. The dose they had used were in the range of human equivalents, so there could be a translation to the human context in terms of dosage. One more drug that we found interesting, and I already mentioned it in the list of clinical trials, is the levamisole. Probably some of you know the complicated story of that drug. It's an antiemetic again, but it was actually approved by the FDA some decades ago to treat colorectal cancer together with 5-FU. It's not used anymore because it's been replaced by other drugs. And at the time of approval, the mechanism of action was quite unclear. They was mentioning immune modulation, but with no further characterization of what was meant by that. The only data in cholangiocarcinoma that came from our research was a case report, which is not very informative, but there is additional supportive data because now it's shown that in terms of immune modulation, it stimulates the Th1 response, which is something desirable in oncology. And there is that trial ongoing in China with data which seems to be unpublished from their own group. The last example I want to highlight is the one is the colchicine example. Again, it's an off-patent drug that has been approved for gout and for other indication. This is a drug that has been extensively repurposed, and in particular, very recently in cardiology. So it's now recommended for pericarditis, and there are other indications coming up. It's a known microtubule inhibitor, but again, polypharmacology is at stake here. The pericarditis indication is based on the very strong anti-inflammatory properties of colchicine. In a rather low impact factor journal, there's a Taiwanese team who showed quite impressive data with the use of colchicine in animal models. They have similar data in hepatocellular carcinoma. The dose they have used is really in the range of human equivalent, so you could really expect a similar effect. I mean, hopefully in human. And this is why they initiated that trial in Taiwan. And as I said, it targets both HCC and intrahepatic CCA population. We had other drugs, of course. One of them was verteporfin and combined with rapamycin, but I think the dose they have used is really too high to be really immediately transferable to the human situation. Outside of our literature screening, as I said, we complemented our searches with additional data we could find. There is the case for the dasatinib, pending the result of an ongoing trial. Then there's also an interesting concept, which is targeting IL-17 in cholangiocarcinoma, because there's a strong association between the IL-17 expression in the tumor environment in cholangiocarcinoma and recurrence. And there are IL-17 monoclonal antibodies available to target IL-17. So they are proof of psoriasis and could actually be used, at least in preclinical model, to test that hypothesis. Extrapolating from data published last year in pancreatic cancer data, extrapolating that to cholangiocarcinoma, there could be a role for MEK inhibition and hydroxychloroquine for tumors with mutation in RAS, RAF, MEK, and ERK pathway. And last but not least, we recently came across that paper in Cell looking at the use of the antipsychotic flowchart, which is also used to prevent chemotherapy-induced nausea and vomiting. This is really interesting work that shows that by giving prochlorperazine before monoclonal antibodies, you can inhibit endocytosis and ensure that the target of those monoclonal antibody remains expressed on the cell surface to augment their effect. And so they have shown this with different monoclonal antibodies and could have very broad application. And I was wondering whether that could not be also relevant for cholangiocarcinoma. They have preliminary clinical data, which is very supportive as well in head and neck cancer. So this is with this, I will go to my conclusion, just summarizing the findings we had. So more than 50 non-cancer drugs have reported some activity against cholangiocarcinoma as of September 2020. Of course, the quality and the strength of the data is very heterogeneous, and there's a need to look into the details to ensure, be sure of the quality of the articles and the experiments being done. What we found out is that there are very few drugs, and not always the most promising, that have moved to the clinic. So I guess it relates to the valley of death in clinical in drug development, and it's probably particularly acute for cholangiocarcinoma, which is a rare cancer. We could find additional drugs from the oncology field, also from having some sort of mechanistic thinking. Of course, there are some limitations of our work. It's not fully comprehensive. We have not looked at all the drugs ever approved. We have not done a systematic evaluation of all cancer drugs, because many approved cancer drugs can probably have a role in cholangiocarcinoma as well. We have not done a full evaluation of the quality of the papers, except for the most interesting candidates that I have presented to you. In terms of perspective, I think, and we are convinced, and I hope I've provided some data to convince you that drugs are tools we can reuse and actually could be one of the only solutions for rare cancers anyway, because of the lack of interest from pharmaceutical companies to develop drugs in rare indications. One important point as well is I think there's really a need, and that's why I'm so happy to have an opportunity to present those data to all of you. There's really a need for a collaborative effort to select the drug candidates that would require additional lab work maybe, or that could move to clinical trials. I think this would be very helpful and useful for the cholangiocarcinoma community to have such a collaborative effort in place. As possible next steps from the Anti-Cancer Fund perspective, and maybe with some of you, that's the intention of this slide, we may want to look into cancer drugs as well. So we could apply a similar methodology to identify cancer drug that could be useful against cholangiocarcinoma, at least that have some evidence of use against cholangiocarcinoma. We could think of a bioinformatics collaboration with some of you, or some of your colleagues. The basic level for me is to look at the relevant targets for CCA and look into existing drugs, whether some of them could hit those relevant targets. A level 2, more advanced, that would require more work and a collaborative effort with some groups with skills in bioinformatics, would do some sort of machine learning. We have such a project ongoing in diffuse intrapontine glioma, and it shows very interesting results. And one of my colleagues, Pan Panzarka, who is based in London, could work together with some of you on similar exercise for cholangiocarcinoma, for instance. Now, I'm the director of clinical research of my organization, and what we want to see is more clinical trials. And I'm a big fan of platform trials, and I know you have such a trial in cholangiocarcinoma, at least in the US, with the batch trial, either in preparation or already recruiting. I'm not sure about the situation. But as a foundation, we would be happy to financially support a new arm of an off-patent drug in batch, for instance, or in any other trial. And in general, because we think platform trials are the way to go to accelerate clinical research in any disease, we would be happy to help setting up other platform trials if you have any such initiative ongoing. And we could provide financial support, for instance. Of course, there are some if coming with that sentence, but of course we in principle are very open to that. With that, I want to acknowledge the support of my colleagues. So you have the whole team displayed on your screen, but here I would like to thank in particular my colleague Pan Panzarka, who is our program director, drug repurposing. But also Rika Capistrano, Clara Rombouts, and Liz van der Borne. Liz is actually on the call right now. They are research managers at the Anti-Cancer Fund, and they helped a lot with data collection and screening of the literature. And I want to thank as well Ilse Roman, who is our program director, pancreatic cancer, and who supervised that work from more a biological perspective. With this is my final slide. I want to thank you. I've put my email address on that final slide if you want to reach out to me after the call. I guess now it will be time for questions. Thank you very much.
Dr. Bouche, this is Donna. There's a question from Dr. Benales in Spain. He says, congratulations for this very nice presentation. Could you please give us your opinion on targeted versus non-targeted therapies for patients with cholangiocarcinoma, the pros and cons of each one? Thanks.
All right. So I have to say I'm not a clinician, so I'm not sure I'm the best person to answer that question, but I do have an opinion. I think we know that targeted therapies can really help, but we know that there are some limitations as well. So I mentioned in my presentation the polypharmacology, and I'm still a believer that some drugs that are not supposed to be that targeted. We call them dirty drug, and I don't like that term. I prefer to use the term polypharmacology. I think that they can have some role, but of course you need to justify that and demonstrate that in adequate models. But I don't have a very strong opinion, and I want to oppose targeted versus non-targeted therapies. Maybe someone else, a clinician, would like to comment further on that question. If not, I'm happy to take any additional question.
Okay. Are there any additional questions for Dr. Bouche? Okay, Dr. Bourad, did you want to close out the meeting?
Absolutely. So thank you, Gauthier, for an excellent presentation. I learned something new every time I hear of your work. I'm hoping that this will stimulate some interactions to take this area and use it to its full potential. So thank you again for presenting. Thank you for everyone for organizing and for the attendees for joining. Have a great rest of the day and great weekend too.
Thank you. Bye-bye.